For today's video lecture, we're going to be discussing the function of the nephron and what happens to filtrate as it travels from the beginning of the nephron in the Bowman's capsule all the way through and out the collecting duct. Our major goals here are, as you can see, goals 14 and 15, which are to define vocabulary with respect to the function of the nephron. So we have a couple words that we're going to be going over prior to looking at the nephron structure. But they're important in order for us to understand how the nephron works. And number 15, explain in general terms what happens to the concentration of ultrafiltrate and why in each nephron location. And I changed that word ultrafiltrate, I changed it to ultrafiltrate from urine, because when we're forming this stuff in the nephron, it's really not urine until it's all done. So you'll see that change from your objective sheet to here. So let's start with a little bit of vocabulary. First off, the word ultrafiltrate is used to describe the fluid that will be leaving the glomerulus, leaving the blood that's passing through the glomerulus and entering the Bowman's capsule. Essentially what's happening there is you're going to have a bunch of fluid leaving the blood, and what you're going to find in there is stuff very similar to blood plasma. However, you're going to be lacking many of the plasma proteins, and we'll talk a little bit more about that. But the ultrafiltrate, basically, we're referring to the fluid that's flowing through the nephron. Active and passive reabsorption are two processes that occur throughout the nephron, and basically they are mechanisms for the nephron to reabsorb certain things from the ultrafiltrate back into the blood. Active reabsorption differs from passive reabsorption in that active reabsorption requires energy, ATP, in order to reabsorb whatever it is it's reabsorbing, whereas passive reabsorption happens as it states, passively. It does not require energy. This is because some things, when reabsorbed, are actually moving opposite their concentration gradient. And if you understand the rules of diffusion, you know things want to move from a higher concentration to a lower concentration. However, sometimes we want to reabsorb things that are in a low concentration to begin with to an area where there's already a high concentration. In order for us to do that, we have to undergo active reabsorption. So you'll see that happening, and I'll point it out as we discuss what's going on within the nephron, versus passive reabsorption, as I said, which doesn't require energy, and you'll see things being reabsorbed that way as well. Excretion versus secretion are very similar. However, excretion refers to a waste product. We're trying to get rid of something. We're excreting urine because it has waste in it that we want out of the body. Whereas secretion is still the release of a substance in the body or out of the body. However, it's not necessarily waste. So for example, your salivary glands secrete saliva. That's not a waste product. That is a substance that we use that is coming from a gland and being put in a different location. Your pancreas secretes pancreatic enzymes, digestive enzymes. Your sebaceous glands secrete sebum, which is an oil. Again, none of them are waste products, none of them are being eliminated. They're all products that we use in the body and that we need. So excretion is more of a get rid of a waste, and secretion is just producing a product. Last but not least, we are looking at three words: hypotonic, isotonic, and hypertonic. And these are words we use to describe the concentration of a solution. Generally, when you have, or two solutions, you have to really have two different fluids or two different areas even in order to use these words. If we had two solutions who were isotonic to each other, that would mean that they both have the same concentration of dissolved substances. So there might be a time when the urine and the blood are isotonic. They both have the same amount of dissolved substances. There's also times when one or the other, the blood or the urine, becomes hypotonic and the other one becomes hypertonic. What that means is, if one solution is hypotonic to the other, it means it has less dissolved solutes than the other. And if one is hypertonic to the other, then it has more dissolved solutes. So hypotonic solutions are very dilute, and hypertonic are usually very concentrated. It doesn't have to be that way, but generally that's how it plays out. Just remember that when you use these words, you're comparing two areas. It would be silly to say that you have a solution that's hypertonic if you don't have it compared to anything else. What is it hypertonic to? What is it more concentrated than? And you'll see a lot of this vocabulary popping up as we discuss the different areas of the nephron. So let's move on now to look at the nephron itself. You have this diagram, I gave it to you in class, and we're going to look at the parts. Hopefully you can identify them, but I'll just run through them quickly again so that we're all on the same page. We're going to start here at the beginning. You have your afferent arteriole with blood flowing in, efferent with blood leaving. The little cluster of capillaries is the glomerulus, and surrounding it is the Bowman's capsule. Then we have the proximal convoluted tubule. Following that is the loop of Henle, which is subdivided into the descending and ascending limbs or tracts, either way. After that is the distal convoluted tubule, and then lastly, the collecting duct. So we're going to examine all of these parts, and we're going to look at what happens in each part and focus on how does the concentration of ultrafiltrate change in these areas, and how are we tweaking it to make it our final product, which is urine. So we're going to start in the beginning here with the glomerulus. So this is your afferent arteriole. Remember, blood from your aorta is essentially making its way in here into this arteriole, and it's traveling through these tiny little blood vessels here called the glomerulus and leaving through the efferent arteriole. And because there's a blood pressure thing going on here, an increase in blood pressure, we're applying pressure to the blood, and we're forcing out the fluids that can fit through the capillary walls. So what you're going to see happening in this area is you're going to see a lot of liquid and a lot of dissolved substances leaving the glomerulus and entering the Bowman's capsule, and that's what it's supposed to be doing. And the kinds of things that are doing that are water, urea, sodium, potassium, chlorine, so you're seeing a lot of ions, bicarbonate, albumin, which is actually a protein, and it's a little unusual that you see it here because generally proteins are too big to fit through the glomerular capsule, but these sometimes a little bit sneaks through, and that's okay. Glucose and amino acids, as well as other things. Now notice that most of these things are all pretty small. As I said, albumin is a protein, and it's a big protein, but it can weasel its way through the capillary. However, large things like cells, red blood cells or white blood cells, should not come through here. Antibodies is another big protein that shouldn't fit through. And when they do come through, that indicates that there's some sort of damage to the glomerulus, because generally the only small dissolved solutes and very small molecules can fit through. So those things will squeeze through and form our ultrafiltrate, and it will collect in the Bowman's capsule and then begin its travel down the proximal convoluted tubule. And what's going to happen here, as you can see, is a ton of reabsorption. I didn't overdo it with the arrows. This is intentional because this is the area, the proximal convoluted tubule, where 80% of what left the glomerulus here gets reabsorbed. So even though we filter almost all the blood through, we reabsorb it back. What you aren't seeing in this picture is that there's actually a small capillary that's going to be running, or capillaries that are running very close to the tubules here. And what will happen is the things that we want to keep in the blood and we do not want to make it to the urine will be reabsorbed out of the tubules back into the blood. And so that's what these arrows signify here. We filtered them out here, and then we said, hey, wait a minute, no, we want that stuff. Let's not take it, let's not let it go out in the urine. That makes a lot of sense if you look here at what is being released and what is being reabsorbed. Some things, for example glucose, unless you have really tapped out your renal plasma threshold, basically the amount that your kidneys can reabsorb, you will always reabsorb all the glucose. Your cells need it desperately. They are going to not let it go to waste. In conditions where you have diabetes, however, you'll see excess because there's literally only so much the blood can hold and the kidneys can reabsorb. But generally, much of the stuff that we lost here from the blood into the filtrate gets reabsorbed from the filtrate back into the blood. Water, urea, sodium, potassium, chlorine, bicarbonate, albumin, glucose, amino acids, all of those things can get reabsorbed. And that's an important thing to note. You know, we have the ability to control what we take back, basically. It's like a little conveyor belt with hands picking up the stuff that we go, wait a minute, I want that, I want that, I want that. Not all is lost just because it sneaks through the glomerular capsule here. What also happens in the proximal convoluted tubule is something known as secretion. So we have the ability to reabsorb the stuff we want, here, but we also have the ability to dump out more stuff that maybe we don't want in this area. So for example, some medications like penicillin that we have, we may want to get it out of the blood and didn't get an opportunity to do it necessarily over here in the glomerulus. So we can secrete it via those same capillaries that are lying here into the nephron. And so these arrows signify secretion of some substances back into the filtrate from the blood, which is like a neat little feature. So we can really tweak what's going on in the filtrate and figure out what we want our end product to be. You know, if your body's dehydrated, reabsorb more water. If it's not, let it go. It's a pretty nice system that we've got. At this point, the fluid osmolarity, also known as the concentration of the ultrafiltrate, is not changing from here to here. And that is because as we reabsorb water, we're also reabsorbing salt. And so that means the concentration is going to stay the same. Now, if we were to reabsorb all the water and leave the salt, it would concentrate. Or if we were to reabsorb all the salt and leave the water, it would dilute. So keep that in mind because you're going to see that this is going to change as we travel through the nephron. So after the distal convoluted, the proximal convoluted tubule, we have the loop of Henle, and we have the descending loop, as you can see, going down, and the ascending loop going up. And what's unique about these, or this area, is that this is where a lot of concentration really happens. In the descending loop, you have major water reabsorption that's occurring. And this is because it is impermeable to salt reabsorption. So the only thing that can be reabsorbed is water. So basically what's happening here is we're sucking up all the water and leaving all the salt, which means as the filtrate travels down the descending loop, it's getting very concentrated. I mean, imagine taking a pot of salt water and just boiling it and boiling it and boiling it until the water is gone. You're going to be left with very, very salty fluid at the end. That's what's going on here. This is happening via passive reabsorption because really the tissue next to the nephron is very concentrated, it's very salty, and so the water is drawn towards it, and that allows us to reabsorb a lot of water. So at this point down here is where you have the highest osmolarity or osmolality of your urine. It's very, very concentrated right there. How did they figure this out? I don't know. I can't imagine how they did these kinds of studies, but it's pretty impressive that they knew it. As you can see, this bullet that popped up here is just showing you that the osmolality or osmolarity, you can look at it either way, is going to increase as you travel down the descending loop of Henle. Now on the flip side, the ascending loop does the opposite. It's going to reabsorb solutes, primarily sodium and chloride. Water is unable to leave, so it gets left in the ascending loop. So now, even though we have a lot of salt here, we're taking a lot of the salt back. And because of that, we're unconcentrating our solution. So basically, the osmolality is going to decrease, and we're going to see that it comes back to a less concentrated state as it's traveling up the ascending loop. And again, as this is happening, you have to remember that we're able to adjust the urine to our body's needs. You know, if we really need water, we really will reabsorb it, or if we don't, we'll leave it in. It's kind of nice that we can do that. By the time you get here, the filtrate is usually isotonic to the surrounding tissues. Here it is hypotonic and the tissue is hypertonic, but after all of this, we've got an isotonic situation, so you don't have any great water loss either direction. Coming from the ascending loop of Henle, we're going to hit the distal convoluted tubule, and that tubule is the fine-tuning area of the nephron. And what will happen in the DCT is you're going to reabsorb some more things. So you're going to reabsorb some sodium, you're going to reabsorb some bicarbonate. If there's a little chlorine there, pretty much anything can be reabsorbed in small amounts here. What you'll also see is you're going to see secretion of things back into the filtrate from the blood, particularly potassium and hydrogen. And this allows us to regulate our acid-base balance, which is a unique feature here. So if you were out of whack a little bit, it would be nice that we'd be able to, look at the blood pH and say, wow, it's really acidic. Let's add a bunch of hydrogen to the urine to help decrease the acidity of the blood. Generally, at this point, the urine is of a lower pH, so it does have a higher acidity, higher hydrogen concentration because we're using this area as a buffer for the whole body. You look at the blood pH, we want to keep it around 7.6. If it's higher than that, we add hydrogen, or we could reabsorb more bicarbonate, which is basic and would allow us to temper that blood pH. So here, as I said, is really the fine-tuning area. The osmolarity is constant. We're not concentrating anything. We're not diluting anything. We're just tweaking. Lastly, the collecting duct is a site of pretty much just water reabsorption. What's important to know about the collecting duct is that it is under very, very strong hormonal control, particularly ADH, which is antidiuretic hormone, and vasopressin. They're the same thing. And what will happen is, as that ADH is released and used, it will cause more or less water to be reabsorbed. So on a normal basis, your collecting duct might reabsorb X amount of water, but when ADH, antidiuretic hormone, is secreted, it will cause actually more protein holes, like pores, to be created in the collecting duct, and that will cause more water to be reabsorbed. So you can understand in situations where you may be dehydrated, you'll see an increase in ADH secretion and a reabsorption of water. So it's an important thing, and we're going to discuss in a separate lecture that aldosterone, angiotensin, ADH, renin system, because it's really important to know how we can use the nephron and use the ability to concentrate, dilute, buffer pH, all of those things to the body's advantage. So it's a pretty important system to look at. That being said, we have all the different parts here. Hopefully you remember the different things that each area does. If not, you can watch this video again. Looking at our objectives, we just wanted to talk about some vocabulary with respect to the function of the nephron, which we did early on, and then explain in general terms what happens to the concentration of ultrafiltrate and why in each nephron location. If you have any questions regarding this, we can talk about it in class. Please make sure that you take the quiz that will be available to you now that you've watched this in Canvas. It will be short and have some general questions for you. And then also please watch the short video regarding the RAA system and ADH. Thanks.